at time, and I think we have a quorum. So welcome everybody to the last CAM colloquium of the semester since next Friday's speaker canceled on us. So I am very pleased that Amy was able to join us. Coming, are you?
All the way over from biotech.
Okay.
Yeah.
So Amy is affiliated in complicated ways, which she will tell us about. But I'm looking forward to hearing about control theory and brain.
Thank you. Awesome. Great. So thank you for having me. It's my pleasure to be here today to tell you about some of the work we've been doing in my lab. So as David said, I am an associate professor. My primary equipment is with radiology at Cornell Medicine. And then I also have an adjunct position here, and I'm in the comp bio department, and apparently also the department of statistics and data science. And then I have a couple field memberships. I have graduate students, but my lab is physically located here. I also have students in New York City that I pull from the neuroscience program. So we're a big mix of eclectic individuals, which you might find reflected in some of the work that I'll be talking about today. So my PhD is in applied math from Case Western Reserve. Some of you had heard this story at lunch, but I went to my first year of graduate school, and I was recommended to take a real analysis class, and I almost dropped out. So I was like, this is not for me, I'm not smart enough for this. I had imposter syndrome, I still have it, it's fine. And then my mom said, just wait one more semester, let's just see what happens. So I stayed, and I was introduced to numerical analysis, like numerical differential equations and numerical optimization and coding, and I was like so excited. I still remember to this day the first time I did the Lorentz equation, seeing the butterfly was like so exciting for me. And so that's what kind of kept me on that track. And I kind of became more applied. During my thesis, I did sort of hepatic models of metabolism using differential equations and stochastic sampling. But then I got really interested in the brain when I came to the medical school. And I remember one of my first meetings with a neurologist. I said, I'm sure you guys understand like about how the brain does this. And they're like, no. And I was like, what about like this disease? Do you understand how that would, no. I'm like, okay, so we have a lot of room for understanding things, especially with the computational aspect, and this is before even the advent of these big publicly available databases that I'll talk a little bit about later, that are enabling sort of machine learning and big computational approaches as statistical modeling to map brain behavior. But let's see, is this going to advance my slides a little bit? All right, so how does the brain work? This is the big question my lab focuses on. And it depends on how the brain works, depends on where you want to look at it in a scale. So here I have sort of all the way on the one side of RNA and DNA, vesicles, synapses. You can look at individual neurons using sort of these really nice imaging approaches, neuropixels. And then we get to more like macro scale voxels, so doing things like MRI and MEG. Then we can look at brain regions, so we can clump these voxels together and sort of get these sort of anatomically or functionally defined regions. And then looking at how those regions interact with each other, sort of at the whole brain network. And then, of course, what I'm very interested in is how the brain interacts with its environment, and how we can map sort of these characteristics of these types of imaging approaches to our behavior in the real world. So again, that's where my lab squarely lies. I have been looking a little bit in this area, but mostly, for the most part, it's been looking at the whole brain in intact in vivo humans. So I don't do a lot of animal modeling. So I always like to talk about the first functional brain experiment, which was done about 140 years ago now. So this Italian physiologist, Angelo Masso, had someone lay on a perfectly balanced table, and then had them think about a mathematical arithmetic equation. And as they thought about it, the blood flows towards the brain more than it does when you're at rest. And all of a sudden, the table kind of tilts in the direction of the brain. So I like to say this is sort of the first functional brain experiment. So trying to figure out what happens when we're thinking these complex and complicated thoughts. And just a mere 110 years later, we have a first functional MRI paper. So this was Seiji Ogawa, who was a Japanese scientist, who first observed the BOLD, or the blood oxygenation level dependent signal via T2 star contrast in protocol in MRI. And what he did was he actually showed a flickering checkerboard to subjects as they were sitting in the scanner. Here's a slice of the brain. If I was laying down this way, it would be kind of like this. So the back of the brain is where the visual inputs are processed. And you can see that when the checkerboard is off, there's very little signal. And when it's on, it's high. And here you can see a summary in time of the off instances and the on instances. And of course, you see blood being rerouted to the visual areas because they're active and they have a metabolic demand. And so then the oxygen is rerouted to that area that is active. So, let's see. Yeah, maybe it was just a little delay, so I won't wait. So there's a lot of different ways now that we measure brain function in humans that are alive and awake. We have things like EEG. The EEG cap you can see here has these electrodes that measure electrical activity. So you can imagine that this may be a little bit more direct of a measuring of brain activity than a blood oxygenation level dependent signal, which is dependent on vasculature. And then there have MEGs also, which have the benefit of really high temporal resolution and high spatial resolution. However, it can only probe a couple of centimeters into the skull, so you can only see cortical activations. fMRI, there's a very high dimensional, very nice spatial resolution, but lower temporal resolution. And on the other end is the EEG with low spatial but high temporal resolution. So all of these sort of imaging modalities to probe the activity and patterns of activation in the brain have limitations and trade-offs. We can also, using MRI, measure brain structure. So maybe some of you have seen them before, but there's these T1 and T2 images that show you the contrast between gray matter and white matter and CSF in the brain, and you can see the anatomical structures. There's also something called diffusion MRI, which allows you to measure the diffusion of water molecules in the brain. Now, if you have white matter, which is the brain's wiring, you can imagine that there are these big bundles of white matter that have very homogeneously organized cellular structures like this, where they're all pointing in the same direction. Then the diffusion is going to happen more in this direction than it will happen in this direction. And then you get anisotropic diffusion. So you can describe the shape of the diffusion using this ellipsoid, and in this case, you're going to have very anisotropic diffusion. It's going to be longer in this direction and shorter in this direction. If you have free water or crossing fibers of white matter, then you're going to have isotropic diffusion or equal diffusion in all directions, and you can describe that with a sphere that has an equal radius in all directions. So you can imagine doing this for every voxel in the brain, or three-dimensional pixel is what I mean when I say voxel, and it's just representing a 2 by 2 by 2 millimeter square area of the tissue, and you can get then these sort of large white matter fiber bundles and sort of these smaller ones by using the anisotropic and isotropic diffusion measures in each of these voxels. So we can get an idea of the anatomical sort of connections that are happening in the brain. Once you have this kind of voxel level information about the directions of diffusion, you can apply a process called tractography that then essentially, here I have a little sort of schematic of it. Let me start my video. So you can see here, this is kind of a 2D slice of the brain where I have one white matter fiber going here and another white matter fiber kind of crossing this way. And tractography, what that does is you start at a specific voxel and you take a step in the direction of maximal diffusion, for example. That's one algorithm. Or you can take a probabilistic step in the direction using the orientation distribution function to sort of characterize the diffusion properties of that voxel. But if you take a step in the direction of maximal diffusion, you'll see you just go along this white matter tract and you reconstruct the fiber. So we can apply this tractography procedure throughout the whole brain and get something that looks like this. And it allows us to estimate the strength of the white matter or wiring connections between different parts of the brain. And of course, there's always limitations in every imaging strategy. Here I just have a video showing you different pairs of gray matter regions in the brain. So these green and orange are different sort of discrete regions in the brain from some predefined atlas, and then the wiring is this blue, sort of the blue wire, or the tractography or the reconstructed white matter between them. And then you can collect everything into this pairwise matrix or structural connectivity matrix that just tells you the strength of the connections between every pair of regions in your atlas. So this is what we call the structural connectome. And then here's just a sort of a visual of all of the different white matter structures that are of importance in the brain. When I started doing sort of neuroscientific and neuroimaging studies, I basically had to beg people for data and promise authorship, and you know, it was fine, but also not great for open science and for big data sets. And luckily, about 10 years ago now, it's actually on the 10-year anniversary of the Human Connectome Project release of data. And so this Human Connectome Project was one of the first of its kind, and it released neuroimaging data and behavioral data and cognitive data and genetic data and familial relationships for about 1,200 people. And so they had functional MRI, they had diffusion MRI, they had anatomical MRI, and they made this data set basically available to anyone who wanted to analyze it. So this is kind of the thing that started all of these big data approaches applied to neuroimaging to try and understand brain behavior mapping. Since then, there have been a lot of other sort of releases of data or initiatives to get data collected. So Enigma is more of a disease sort of data set initiative that tries to combine data from clinical studies that were performed across different institutions, which I'm sure you can anticipate big problems with, like combining data from different centers, but it's at least a push in the direction of trying to make bigger data sets out of clinical data, which are generally usually kind of small. And then there's some things like the adolescent brain cognitive development data set, which is a very huge undertaking. It's about 10,000 individuals, and they're studying them from ages 9 to 14, so they're going to be about 5 years of data that they're every year scanning these subjects and getting their behavioral and genetic, and even have like actograms and all of these things that are really a lot of quantitative data on behavior of humans and their anatomical and physiological structures in their brain. Okay, so as I talked about the functional sort of analysis that we look at, we can use fMRI to sort of extract functional time series of different regions of the brain, and you can kind of relate you can make a functional connectivity matrix, which allows you to sort of assess the strength or the co-activation patterns of two regions together. So if two regions fire together always and deactivate always, then they're going to have a very strong functional connection, we call. And then the structural connectivity I already talked about, that's the wiring. And of course, if two regions are wired together, you might think that they fire together. And if two regions are not wired together, you might think that they're not correlated, but that's not the case in the brain. So it's a lot more complicated than that. And so there's lots of different approaches: mathematical, statistical, machine learning, these differential equations, linked neural mass models, graph theoretical models that attempt to map function and structure to each other. And today I'm going to be telling you about some of the recent work in our lab. All right, my advancer isn't working so well. That is focused on network control theory. So we have a lot of different results related to this network control theory approach. So the choice to use this network control theory to map structure and function and model brain dynamics is driven for myself for both theoretical and practical motives. First, the theoretical, so we know that the brain is a complex system and that we know that it can be controlled because we control it every day. So our neural systems change to meet the dynamics of our environment and our task demands, right? So it allows us to, let's say, pick up a laser pointer or walk from here to there or perform some cognitively complex task. So we can control this complex system that is the brain. And more research is allowing us to see a window into the neuroimaging substrates of this mechanism that we call cognitive control. And there's an engineering analogy then to this cognitive control. So the state of a complex system can be modulated by some energetic input. And the energetic input allows the system to go from state to state, traveling along these trajectories in the dynamic landscape. So here you can see an example of that, where there is some state 1, let's say that's walking, and state 0 is, let's say, standing. So you have this sort of trajectory of brain activity that takes you from walking to standing. So there's an analogy in this engineering sort of framework in network control theory. And so the brain is sort of this interesting example of a complex system with a defined pattern of interconnectedness. So that's the structural connectome that I was telling you about before, where they have nodes in the brain, these gray matter regions that we delineate with some kind of atlas, and that allows us to measure the sort of structural connectivity between them. So we do have this interconnected network, and the brain activity at any given point in time is the sort of brain state that you're in at that point in time. Then the practical. I'm a very practical person, and so I abhor throwing away data. And a lot of times what people do, so this is an example of a time series. So the time is in this dimension, and in this dimension is space. So these are all voxels in our fMRI. And you can see, let's just ignore for now these like black things because that's where people are deep breathing. So there's definitely artifacts and issues with the time series itself. I'm not going to get into all of that, but above you have the gray matter voxels, or the areas of the cortex and subcortex, and then here we have white matter, which is obviously much more noisy. Generally, people throw away white matter and ventricle, and they just look at gray matter. And then what they do is they compress in space and time. So the spatial compression happens at the level of the atlas, where you have a bigger sort of cortical region that you can average the voxels within to get a time series for just that region. And then they compress over time to get a functional connectome. So compressing in time here, I just mean that you look at the time series over the regions and then you correlate them, let's say, to get a strength of functional connectivity. So in the end, you go from this 100,000 by 1100, let's say, in time, to a 268 by 268 matrix. So there's a lot of data that's being sort of lost in this compression. So if you use network control theory, you can maintain that temporal dimension. You don't have to compress over time because we are looking at state dynamics. So we are looking at each point in time and looking at the activation of each point in time. So here is just a kind of visual example of what I mean. So here we have each session for each subject. So let's say there's 5 sessions for 47 subjects. You have the regions by time. So we've already compressed it in space to get our regions. And then we have each region's time series. And then we can apply, let's say, a very simple approach is k-means clustering to identify these commonly recurring states of brain activation. And then when we have these states, we can reconstruct for each subject in session the sort of state temporal time series. So how each TR is assigned to a specific state. And then for each subject, you get a vector that's the length of the time series that tells you which state that particular brain pattern was assigned to over time.
What are you actually measuring at each?
The brain activation. So it's like the regionals. So let's say you divide the brain into 68 regions. So it'll be a 68 by 1 vector at each time point. That's representing the amount of activation. I'm saying activation.
Measured by oxygen?
Yeah, it's the blood oxygenation level dependent signal. So some people have applied these approaches to MEG, which is neural activations for electrical and interferes with magnetic field, so you can measure it. Or EEG, which is actual electrical activation. But the bold is always like a little bit of a question mark because you're measuring vasculature. There are lots of studies that show there's a tight coupling, at least in healthy control individuals, between vasculature and neural activity. So the brain is this very super cool thing that I've devoted 15 years of my life to. But anyway, but it is this very super cool thing that has been designed to really be able to reroute metabolic substrates where there is demand. So it's a really responsive system. Okay, so now let's say we have those 4 states. So here's an example from, yeah.
Sorry, you're thinking of a state as being functional region and some behavior in time. Is that behavior in time quantified as a time series?
Oh,
and that's what you're doing?
No,
k-means on,
okay,
or
so
you're doing
most
it on
of
a Fourier
the
spectrum?
analysis we do is in resting state. So they're just sitting in the scanner, not doing anything.
Okay.
That's also another point that I could go on and on about because who knows what somebody's doing. They're like, people are told not to think of anything or do anything in particular when they're sitting. But who knows what they're doing. And actually there's some really interesting work in decoding internal mental states during rest. And there's some interesting work in decoding fatigue and sleepiness that occurs. You can see it kind of increasing as the scan goes on. But for the most part, the analysis we're doing is on resting state, which should be some kind of uncontrolled dynamics. There's, I don't know if you've heard of like the default mode network, but it's these regions in the brain that generally fire the most when you're told to just sit in the scanner, but without doing anything, and they are associated with autobiographical memory and self-referential thinking. So you're kind of thinking about where your body is in space, and maybe you're thinking about what you have for breakfast or whatever. So yeah, you're told to not move. So there's hopefully no confounds of like motor and visual. You're told to like stare at a crosshair. So there's hopefully no sort of activation, not much activation differences in your visual network. But there's a lot of like self-referential memory and sort of autobiographical memory. Yeah. So once we have these sort of vectors of state dynamics, then we can calculate things like fractional occupancy of states, so like how percentage of time spent in a specific state. We can calculate things like dwell time, like how long we spend in that state once we transition to it. Appearance rate, so let's say perhaps the number of times you transition to a state in over a minute's time. And then you can calculate things like transition probability, so how likely you are to go from one state to another or stay within the same state. And this work specifically that I'm telling you about right now, I'm not going to do that first. Oh yeah, first I want to talk about control energy. So maybe some of you know this better than I. But so this is one of the first papers to look at network control theory applied to mapping brain dynamics. So here again, this is what we've been talking about, where we split the brain into this atlas, we do fiber tracking, we get a structural network. So now we have a structural network, and then we have these functional brain states. So then we can calculate using network control theory, and assuming that this is kind of a linear time invariant system that we're modeling. So here we have dx dt, and then we have A, which is our structural connectivity matrix, so it's the network represented in black here. And then x of t is the activation vector across every region of the brain at time t. And then you have some control strategy contained in B, which I'll talk a little bit more about later. And then some external input to the system, U of t. Okay, so it's a linear time invariant system. And we can apply, so this is kind of a schematic of our idea about transition energy. So the idea behind this transition energy is we want to calculate the amount of energy that's needed to inject into the system to complete a state transition. And if you look back at that time invariant system, let's see if I can get to the next. Yeah, this is what I should show. So we have this linear time invariant system. You can see that this dx dt equals a of x of t is just network diffusion. And so that's uncontrolled diffusion. So let's say this light gray trajectory is taking us from our initial brain state through the dynamic state space into this sort of x of t, which is uncontrolled diffusion. So what would we need to do to control that trajectory to get it to the state that we want to observe? And in this case, it's those states that we had defined using K-means clustering. And so here, the control strategy, which is sort of a matrix that represents the amount of control that each region has in the system. So you can assign more control to some regions over others. So a lot of times we'll start with the uniform leading and say that every region has the same control. But of course, we might also want to incorporate something about pathology or receptor density to allow us to investigate how spatial weighting might inform that transition energy. So essentially, the network control-based state transition energy allows us to calculate sort of the amount of energy that's needed to shift away that uncontrolled diffusion trajectory towards the final state that we would like to achieve. Yeah?
Are those A and B matrices similar across different human subjects?
Oh, that's a good question. So A is, they're pretty similar. So generally, if you don't have a disease like MS, or you haven't had a traumatic brain injury, or you don't have, a split corpus callosum, then generally they're pretty similar. Although I will say there's some really cool studies showing changes in white matter integrity fibers, like from motor training. So there's this really cool study where people did juggling training, and they showed that their white matter connections in a certain region were actually improved when they learned how to juggle. So yeah, the properties are similar, but you can have genetic and environmental and kind of characteristic differences. Yeah.
Are we thinking about this as an open loop system, or
Yeah, that's a good question. I wouldn't say it if it was, I would say, I guess in the intact human just sitting there, yes.
Okay.
Yeah, yes. Yeah, like the question, I think what you're asking is like, is this like an external thing? I guess if you're sitting there and you're changing what you're thinking about, the external control might be your own volition. Do you know what I'm saying? But you can also think of this in terms of like external stimuli, like deep brain stimulation or transcranial magnetic stimulation, or even visual stimulation that causes some activation to be injected into the system. But that's a good question. It's probably more philosophical. Yeah.
Sorry, when you say control theory, I want to know who is controlling and what. Is the idea that the evolution somehow selects a good bee to help us get the signal to the state where you want it to be, given the set of inputs u of t? Or is there a control u here and we're the ones controlling? I cannot.
That's a
figure
good question
this out.
because I don't think that we know the answer to that. And there's different approaches that you can take to sort of analyze different aspects of the system. So I'll give a couple of examples later, but this B, like I said, it can either be uniform or weighted. So this B might contain information about some specific hypothesis that you want to test. So if I want to test the effect of probing, let's say, dopamine in the brain, if I flooded the brain with dopamine, then the regions that have higher dopamine receptors would probably have more activation and more controllability over the system, right? Or I could be thinking about in terms of B, in terms of injecting signal into the system with like a TMS oil or something like that, where there's one region that I'm injecting the system, the control through. So it depends on the type of thing that you're trying to model, I would say. But in sort of uncontrolled, like sort of resting state, we normally think of this V as uniform, so we just say every region has equal control, and then the U is sort of, I guess you could philosophically say somehow your free will is sort of pushing your thoughts into a certain direction. Does that make sense? Hopefully. That's
Yeah.
kind of how I thought about it in the past.
I guess I'll need to see the examples to fully
Yeah,
get
maybe
my
it would
head around
help.
this.
Yeah, maybe it'll help if I get to the examples.
So I'm wondering, X of t, each component of X of t corresponds to a region, right?
Yes,
How about the components of U of t? What do they correspond to?
those are also regions.
So every region has some sort of control
Yes,
of it.
yes, exactly. And and so B is usually an identity matrix, but we can assign weights to the diagonal as well. And you can like zero out control for some regions if you think that, you know, they shouldn't have control in a certain scenario. Yeah.
So if we do not control the system, do we have some interpretation for the equilibrium state?
Death? I don't know. Yeah. So I've thought about this a lot too. Equilibrium state wouldn't make sense physiologically, I think. So the brain is all about balance. So if you have, like, let's say a stimuli that stimulate your amygdala, like a lot, if you take away that stimuli, your brain will bring the activation level of your amygdala down below normal resting state activations, because it's trying to balance out things. So I wouldn't say there's a stable state unless it's zero everywhere. Yeah, hopefully. I mean, so there are some interesting work. There's some interesting work in meditation, and then I'll get to psychedelics at the end, which might be like something that's another philosophical problem. But but the meditation state is kind of interesting because that does seem like there is this overwhelming coordination of brain activation throughout the brain. So I don't know if that's also could be, yeah, you could think of that as like sort of a stable state. But, yeah. All right, so here we have our linear time invariant system, and then some of you I know have done network control theory before, so I won't belabor the point, but this is our controllability Graumian. So then the transition energy that we're talking about specifically here is is sort of the amount of energy you need to take that uncontrolled diffusion and get to that final state, considering this the controllability of the system. All right, so now I'm going to talk about some applications which hopefully will illuminate more about the theoretical parts that I just discussed. So first I'm going to talk about network control theory in healthy populations. So one of the very first papers to apply network control theory in human brain data was from Danny Bassett's lab at UPenn. Who's like a fangirl for Danny Bassett. I love her, she's great. They found that regions were more structurally connected to the rest of the brain also had more control over the system's dynamics. So I guess that's not too surprising, but it's like a nice validation of the method and application. And then they also found that regions in the default mode network, which is again the region that's sort of active when you're just sitting in the scanner, not doing anything, and the somatosensory networks had a larger portion of that available control in the system. So the default mode network and the somatosensory networks are sort of at opposite ends of the spectrum of the hierarchy. So the somatomotor network sort of deals with your environmental actions and your incoming signal processing. And then, of course, the default mode is this higher order sort of self-referential autobiographical memory region. So it's kind of interesting to see that the opposite ends of that hierarchy have more control over the system. And then there was another paper recently, this is 2020, this is Human Connectome Project data. So again, it's a 1200, N equals 1200 data set of fMRI and structural connectivity. They applied k-means clustering, and then you have these five commonly recurring states of brain activity. And they analyzed their state dynamics and transition energies across rest and task. So the HCP data also has like a task in the scanner, which is sort of a memory task. And I don't know if anybody knows about this, but like 0, 1, and 2 back are like sort of increasing in their amount of memory that are the amount of cognition and memory that are needed. So you can kind of get a continuous measure of cognitive demand across those tasks that were done in the scanner. And so here below is just a sort of summary of the state centroids in terms of their cosine similarity with these kind of canonical networks. So I won't get into too much detail, but DMN is default mode network. Frontal parietal is like a sort of cognitive control network, or executive attention network, and then the VIS is more like the visual system. So you can see these the higher order of the hierarchy and the lower order of the hierarchy. And then they showed that this one on the bottom right is the transition probability matrix, and they showed that the highest probabilities are from visual to default mode network states higher to lower order, and then the lower probability for transitions from higher to lower. So there seems to be an asymmetry in the transition probabilities that are favoring bottom-up transitions. When I say bottom-up, I mean from like lower order networks to higher order ones. Yeah. And then of course here you can see these are the fractional occupancies of the 5 states, and rest is in blue, so that's that one where we're just sitting in the scanner. And you can see the default mode network has a higher occupancy and the visual has lower. And then for the end-back task, which is that memory task, we see the default mode is lower. This is also called the task negative network. So it means when you're doing a task, it deactivates. And then the visual is higher. So it seems like there's a shift in the fractional occupancy of these states when you have different cognitive demands. And then, oh, sorry, I didn't type that, but it's okay. So then below you have the fractional occupancies across the 5 states for the different rest, 0, 1, and 2 back tasks, which are again increasing cognitive demand. And you can see there's kind of a monotonically increasing fractional occupancy of these VIS states and a monotonically decreasing the default mode network states. And then the transition probabilities, of course, look very different between the rest and 2 back, where you have more transitions into the VIS, which is kind of going hand in hand with higher fractional occupancy. And then they even mapped the sort of transition probabilities to performance, so how well you did on it. And it seems if you have higher probability to transition from this VIS negative to a default mode network positive state, you do worse. But if you go to the state that has to do with sort of executive attention, then you do better. So it does seem like there's even a performance correlate of the state occupancy and probabilities. And then they calculated this sort of control energy and correlate for every pair of states. You can calculate this sort of transition energy between pairs of states, and then they correlated that with age. And they actually found that for every state 2, this default mode network state, which again is the autobiographical state, you have a negative beta map, which means the older you are, the easier it is to slide into that autobiographical state. I'm 42 now, so I can understand that. And then there's this really cool paper by a collaborator of mine, Lyndon Parks, that was just published in Science Advances, where they're looking at the transition energies across development. So they had this really cool data set of about 800 subjects that are aged 8 to 22 years. And they used this sensory-fugal axis of cytoarchitectonic similarity that allows you to sort of arrange things higher to lower order, as I kind of talked about before. And then they calculated transition energy required to go top-down transitions and bottom-up transitions, and then they looked at the asymmetries of the energetic demands. And their main findings were that the transition energy is greater for transitions going from top-down than from bottom-up. And then relatedly, if you do uncontrolled dynamics, They preferentially flow up the cortical gradient to cytoarchitecture, so they go from lower order to higher order within uncontrolled trajectories. And then they also found a negative correlation between transition energy and age. So transitions seem to become easier throughout development. And the age-related decreases in energy were more pronounced for the top-down transitions, indicating a reduction in bottom-up versus top-down asymmetry across development. So it might become easier for people to control their bottom-up signals if they have an easier time transitioning from top-down. So this is a student of mine, Elaine, who did her senior honors thesis, and this is data set. It's a really beautiful data set. It's from a single female subject over 30 days. She had an fMRI scan and a blood draw each of those 30 days. It's called the 28-week data set. And so we have then her hormone levels at all points across this menstrual cycle. And we see the estrogen and progesterone here across the 28 days. So we took their fMRI data, we did the clustering, we found these 5 states. We calculated transition energies between every pair of states, and we actually found a correlation with estrogen. So there seems to be a higher energetic demand for transitions as estrogen increases, and a lower entropy of brain activity dynamics as estrogen increases. So this was kind of a short senior thesis project that she's now working on expanding into a little bit more in-depth analysis of that data. It's a beautiful data set. Okay, so next I'm going to talk about neurological disease and injury. So this was from my PhD student who just graduated, Emily Olafsson. We looked at the evolution of post-stroke brain dynamics. So one of the times that I was telling you about before, where I sat down with a neurologist, I was like, oh, you guys probably know how the brain recovers from injury, and they're like, no. So I think this is one of the main thrusts of my research, is trying to figure out how brains reorganize in order to compensate for injury. Like, if you have a damage to a specific part of your brain, how do you then compensate for that damage to become functional again? Because if we can understand that, then we might be able to support those mechanisms and get people recovering faster or better. And so here the number of subjects we have is about 23, and we had 5 sessions after the stroke. I think the sessions were 7, 14, 30, 90, and 180 days after stroke. So it's a first-time pontine stroke. You can see the lesions here. This is kind of a heat map of the lesions in the brain. It was a pontine stroke. And then these subjects suffer from motor impairments. And so then you can see the color bar here is the motor scores, where higher values mean better motor scores, and green values mean worse motor outcomes. So here what we found when we did the brain states analysis was that we found that stroke subjects spent significantly less time in a state that was dominated by frontoparietal activity compared to the controls at all time points. And then we also found that the stroke subjects were less likely to stay in this FPN state and less likely to transition to the state, so it's not surprising. But interestingly, we found that the fractional occupancy and dwell time of this FPN state was associated with better outcomes, but only in individuals with a dominant cortical spinal tract damage. So if you're right-handed, your motor strip on the left side is the thing that controls your right hand. And so if you're right-handed, the motor representation is larger on your dominant side. And on the opposite contralateral side, there'll be less motor representation. So what we think we're finding here is that when people have a dominant CST damage, they're already primed for better recovery because part of their brain, which is more like frontoparietal, is already devoted towards motor recovery. So we found that when the dominant CST was damaged, the more time a person spent in this frontoparietal state, the better outcome they had. So it seems like that might be supporting this idea. And that goes towards personalized treatment of stroke outcomes. So if you have a dominant CST that's damaged, it might be better for you to be in a state that is in that frontoparietal state, rather than if you had a non-dominant hand that was damaged. We also found that there was a correlation between the change in dwell time of this FPN state and the lesion overlap with the dominant CST. So the more there was an overlap with the CST, the more there was time spent increasingly over time in that FPN state. So we think that we think that this could be used in a context for sort of non-invasive brain stimulation, which could support activation of this FPN network in people who had dominant CST damage. So the next study was by my postdoc, Jared Toslu, and this was in studying brain activity landscape in 100 individuals with multiple sclerosis. So multiple sclerosis is a disease that can impact white matter connections, and you get these lesions that cause damage to the white matter connections, and you have overall higher inflammation. It's an autoimmune disease. And when we applied the sort of network control theory, well, state analysis at first, we found those 6 states on the left side, and we calculated the transition energy matrices for every pair of those 6 states. And when we used that transition energy matrix to classify into 3 groups: controls, people with MS with disability, and people with MS without disability. And when we looked at those model feature importance coefficients for the classifiers, we observed that the people with MS without disability tended to have decreased transition energy compared to controls, and that people with MS with disability tended to have increased transition energy compared to controls. So we interpreted this as perhaps some sort of compensation mechanism that initially keeps that disability at bay, but then is exhausted over time and results in disability of the individual. So this could be this, and this is not the first, we're not the first people to propose that there's a compensation mechanism that occurs early on in the disorder of MS. We also found that regional entropy was largely but not significantly different between the 3 groups, but there was a global trend for decreased entropy in MS compared to controls, and increased entropy in people with MS without disability versus those with disability. And we found a significant correlation between decreased entropy being related to increased energy and increased lesion volume. So it seems like the more lesions you have, the harder it is and the more energy demand to transition between these states. And so this sort of results might tell us about the possible mechanisms of how MS-related damage to the brain structural backbone might impact brain energetics. Okay, I don't have much time, so I'm going to skip past, this is traumatic brain injury, role of dopamine. I want to make it to the psychedelics part, which is going to take me some time, but I will touch on this. This is substance use disorder, which we believe could be due to an underlying dysfunction of dopamine in the brain. And so we had this idea that if this dopamine, we did this simulation study showing that if you simulate dopamine dysfunction, you get increased global energetic demand. And so what we then hypothesized was that perhaps in a population where we think there is a dopamine dysfunction, that there would be higher energetic demand in transition between states. And so we actually found that that was true in our substance use disorder group. This was from Human Connectome Project data, where I said before that they were all controls, but some of them also have diagnoses of addiction disorders. And so we looked at the sort of global transition energy, and we saw that the transition energies across the board, the global values were increased in substance use disorder compared to healthy controls. And that more time was spent in the lower order states versus the higher order states in substance use disorder compared to controls. We also found that the bottom-up transition energy increases and their top-down transition energy decreases in SUD, which sort of is shifting those dynamics to prefer those lower states than the higher states, which kind of goes hand in hand with what we saw with the previous fractional occupancy study. And then we also showed that regions that have a higher transition energy, so we also have this method to calculate regional transition energies. So regions that have higher transition energy in SUD also have lower entropy compared to controls. Yeah, I want to get to the psychedelic work. All right, because that's the kind of most on topic sort of thing. Because I'm sure a lot of you have been reading about psychedelics and their potential uses for mental health disorders, and even now I'm hearing about neurological disorders like disorders of consciousness and traumatic brain injury being a potential thing to treat with psychedelics. And you can see here the number of clinical trials with psychedelics has kind of really exploded in the last few years. And if we're going to be using these psychedelics for clinical treatments, then we all should maybe understand the mechanisms about how they work and how they affect the brain activity and brain structure. And so these neuroimaging studies, there's been about a decade of them actually in psychedelics, mostly out of Europe because it's too hard to do in the United States. But one relatively consistent finding is that the brain activity demonstrates an increase in entropy under psychedelics. And there's this relaxed belief under psychedelics, or Rebus model, that proposes that perception and belief are shaped by both prior knowledge and incoming information. So if I believe that this wall is not going to move, and I have a little bit of sensory noise that tells me it's moving, my prior is going to say, no, it's not moving. But if I have a removal of that prior, then the sensory noise coming in could be converted into some belief that is not aligned with what I know to be true. So that might be another reason why people hallucinate very strange things when they have psychedelics. So it also has reciprocal or enabling implications for bottom-up information flow. Like I said, those incoming information signals may have more dominance in the perception. So it's theorized that the increase in entropy might be reflective of the reduced energetic demands for the brain to navigate through its dynamic landscape. And so we wanted to test, this is, by the way, this is an entropy increase under coffee. So it also has a similar neural mechanism, not just but it does increase entropy very much. So this is work that was done by my student partner Singleton, who just graduated with his PhD. And we hypothesized that perhaps the psychedelics observed effect in increased entropy is concurrent with the flattening of the landscape. And so here is just an illustrative sort of flattening of the landscape that we propose under psychedelics. So then it would mean that the transition energy would be greatly decreased. So I'm not going to get into the details of this, but essentially there was a study by a collaborator of ours, Robin Carr-Harris, where a person took LSD and then they sat in a mock scanner for a while to sort of get them acclimated to that environment, and then they went to the scanner about 2 hours after they were taking their LSD to be scanned while their eyes were closed and they were quietly sitting in the scanner. There were 20 healthy volunteers that did this, and one of them was excluded for anxiety and 4 for excessive motion. So it left us with 15 subjects. But we also had placebo for them. Placebo. Let's see right here. There's no placebo for a psychedelic study. That's one of the problems in it. But anyway, and then we had another study where we gave them IV, I didn't, but they gave them IV psilocybin and had them go into the scanner and had an fMRI. So the first thing we noticed was the fractional occupancy under LSD and psilocybin. They were spent more time in this somatomotor state than the frontal parietal state, which is more of a top-down. So they had more occupancy of these lower order states and less occupancy of these higher order states. And then to test our main hypothesis, we see that LSD and psilocybin both lower all state transition energies. So across the board, all pairs of state transitions have lower energy in LSD and in psilocybin. And you can see this is a very nice illustration. So this is the time series of the transition energies, and each color is a person. And you can see here, this is the placebo infusion. And there's like a small dip in transition energy because we've also realized that transition energy is related to arousal. So if a person's getting an injection, they're being told they're about to get an injection, and they don't know if it's going to be LSD or psilocybin, you can imagine you might be on high alert. And then you realize it's placebo and you start to relax again. But then the psilocybin, you can see there's a great reduction in the amount of energy needed to transition between every pair of brain activation states. So across the population, there was also a positive correlation between the amount of reduction in energy and the reduction in dwell times of these states, and concurrent with the increased appearance rate. So it seems that, in fact, the entropy is increased. So we do see that across the population, again, the amount of energetic decrease is related to increases in entropy. So here we just did a simple lump-of complexity analysis of the brain state time series. And we were very happy when a collaborator of ours in a totally independent data set correlated the levels of complexity calculated from EEG data, so no longer do we have to worry about the vascular components of psychedelics, and psilocybin level. So they actually continuously monitored the psilocybin level in the blood, and they saw this positive correlation. So the more psilocybin there was in the, or psilocin there was in the blood, the higher the entropy of brain activation. So we wanted to look at by what mechanism did these transition energies decrease. So we know that LSD and psilocybin work on a serotonergic receptor, and specifically they mainly work on the 5-HT2A receptor, which is a specific serotonergic receptor in the brain. And that has a specific brain distribution. So there are some regions that have more 5-HT2A receptors and some that have less. And so what we wanted to do is see if we shifted that uniform control weights and we use a control strategy that now is related to the amount of 5-HT2A receptors, what happens to this transition energy? So again, revisiting our result, these are the reductions in transition energy we see under psychedelics. And then we used our placebo centroids and we calculated what would happen to the energy if we used a control uniform strategy, this unweighted, versus a 5-HT2A weighted strategy. And you can see there's a reductions across the board in the transition energies needed to go from state to state when you give more control to regions that have a higher 5-HT2A receptor. And then to be a little bit more robust, we use a spin permutation test, which preserves the autocorrelation of the regions in the brain, and we've still found largely reductions in transition energies. We also compared the 5-HT2A serotonin map against other serotonin maps, and we see that compared to the other serotonin maps, this 5-HT2A receptor sort of spatial distribution seems to be optimally constructed to lower transition energies in the brain. So this is the sort of most recent work we've been doing. This is DMT, which is a fast-acting psychedelic. So the person went into the scanner, they were in there for 8 minutes, and then they were told they were getting an injection, and they either got placebo or DMT injection, and the hallucinations start almost immediately, the effect, and then about 10 minutes or 15 minutes after the injection, the effects wear off. And so we had this really nice data set with which to test in a dynamic way the amount of energy reductions. So here on the left, we're showing the time-resolved transition energy. So this is kind of like a transition energy from subsequent time points in the fMRI time series. And you can see that the red and black, the red is placebo, the black is the DMT, and before the injection, they're about the same. But then as the injection happens, people become on high alert with the placebo, then they realize they're in placebo and they're okay again, but then the energy demands remain depressed and kind of continuously go back up after the effects start to wear off. We also see that the change in global control energy between DMT and placebo, that's the sort of blue is the control energy from the fMRI. And they also had simultaneous EEG, so they also had EEG cap, poor people. EEG cap and an fMRI scanner undergoing DMT, it's got to be pretty tough. But then they also had the signal diversity measurements, and we see that there's a tight coupling between the energetic decreases and the energy, or the signal diversity increases measured in the EEG. We also sort of did a time windowing average of the control energy in the fMRI and compared that to the signal intensity, or the subjective ratings of intensity, and we see a tight correlation there as well. We further again wanted to look at regional level characteristics to see if there was some special contribution of 5-HT2A receptor density. And so here you can see the regional distribution of the nodal control energy differences between DMT and placebo, or pre versus post. And you can see across the board there's mostly blue, and that actually we wanted to compare that with the serotonin receptor density, so I'll show that in a minute. The middle plot here is the DMT nodal control energy correlation with EEG signal diversity. So that means that for each region, it's the correlation between that region's control energy and its signal diversity. So the negative values indicate higher energy is related to lower entropy. And then the far right shows for each region the correlation between the region's energy and the subjective drug intensity. So negative values there indicate the region's lower energy was related to more intense drug effects. So then we correlated each of these regional metrics with the 5-HT2A receptors, and we see negative relationships across the board. So it seems that those regions that had larger, let's say larger changes in control energy also had nodal control energy as temporal correlation with signal diversity and drug intensity are associated with the serotonin 2A receptor spatial pattern. And then we did a dominance analysis, kind of comparing the different serotonin receptor maps, and we see that this 5-HT2A appears to be most associated with those statistics of that I described on the previous slide. So let me wrap up with this last simulation study. So there was this paper from 2022-23 where they did a study and they modeled plasma DMT concentration over time. And so we had this idea of what the trajectory looks like, the concentration of DMT. And we wanted to simulate what would happen if we also looked at considered the temporal dynamics of the DMT as well as the 2A receptor density over the regions of the brain. So now we have this time-varying control strategy where we're incorporating both the plasma concentration levels of the DMT and the receptor density. So we again revisit this linear time invariant system. And we use the placebo-bold fMRI time series, and then for the control strategy at each time point is then now this vector from this matrix here. And we apply that to the placebo time series, and we see that there's a tight match between the simulated DMT, which is again the placebo with that applied control strategy, and the empirical DMT. So this tells us that using information about the temporal dynamics of DMT and the spatial distribution of the receptor that it impacts can give us an idea of the transition energy time series. Full disclosure, this was created by ChatGPT, I think. So I think Parker did that, so it was pretty interesting. So the brain is a complex system, and we can use network control theory sort of as a unifying framework to combine information about structural connectivity and functional activation patterns and receptor information to understand the sort of dynamics and the effect of different diseases and disorders on these dynamics. We can also use it to sort of look at the effects of cognitive tasks or hormones or pharmacological agents on brain dynamics, and look at the effects of neurotransmitters on brain activity as well. And we have shown that there are global energy increases in these different diseases and disorders. I didn't go into this exactly, but we also have seen an increase in transition energy in subcortical structures of children with family history of substance use and estradiol levels. So this might give us a better idea of the mechanisms of some of these disorders and diseases. And we also know that there's this large energetic decrease in acute psychedelics, which could point to their therapeutic applications in depression and anxiety and different diseases and disorders. So we might be able to then use this information to develop treatment strategies that allow us to push the landscape into a direction that would be beneficial to the person. So with that, I just want to say thank you to my funding sources in my lab.
Questions on the way, but time for more. Max?
Sure. So I kind of want to make sure I understand what you're doing. So maybe, for instance, with the psychedelics one, is it that you learn A and B from scans of the model, and then you determine your energy? Is that like kind of the flow?
Yeah, so the A, in the case of the psychedelics, we unfortunately didn't have diffusion MRI. This data was handed to us from a collaborator, again, because it's like impossible to do these types of studies in the US. It's getting better, but it's sort of impossible. So we didn't have diffusion MRI, which is usually what we use to extract the A matrix, which is the structural connectivity matrix. So what we used was a placeholder there. We used the same structural connectivity matrix for everybody that was extracted from a large data set, the HCP data set. So all of the differences we're seeing in that particular study are from brain activity. There's no there's no assessment of whether the structural connectivity matrix A is different between the psychedelics and the placebo.
So
That's
when you
fixed.
learn this energy, you're learning it directly from the signal. You're not learning.
Yeah, it's from the activation, from the dynamics of the brain activation.
So is your underlying model that you're imagining happening, that when you inject the drug, the A and B change?
No, I
Or
think I don't think A is
is the
there.
drug you?
Yeah, so I think the drug would be contained in B. That's our idea, right? So our idea is that B, which is the control strategy of each region in the brain, it's how much each region controls the dynamics of the system. When you give a psychedelic, it preferentially activates regions that have more 5-HT2A receptor. So that we kind of think could mean that it has more control over the system because it's more active. So that B is what I'm thinking is changing. And from what we saw with those kind of simulation studies, when you change the control, the amount of control each region has according to the amount of 5-HT2A receptor, you get a flattening of the landscape.
And I have one more thing. So it seems like the general statistic of the energy is pretty informative. I mean, I think that's most of what this talk was. When you do like multiple, say if you did like 2 trials on the same person, and you learned A and B, do you get kind of the same thing?
So if you're talking about test-retest reliability, I actually did an assessment of this because functional MRI is notoriously noisy, and it can change, like so many things influence it. The amount of coffee you've had, the amount of sleep you've had, how sleepy you are in the scanner, what you're thinking about in the scanner. So functional connectivity is very noisy. So I compared the test-retest reliability of functional connectivity to the transition energy. And the transition energy is much better. So the test-retest reliability there, like a correlation between the 2 different observations of 2 different scans was like 0.8. And for fMRI, it's like 0.4. So you get like a 2x amount of reproducibility there. And I'm thinking it's mostly because that transition energy considers the structural connectivity, which is more static and less dynamic.
Thank you.
Yeah.
Jackson.
So how do you go about training your A matrix, and is there a reason why your system is well modeled by a linear system, or do you know if it is?
So I think that goes back to my slide I had up there about all of the strategies that people are trying to use to map structure and function. So the idea behind network control theory is that activation flows through from region to region according to the sort of difference between the activation of two regions and the amount of connection between them. So if there's two regions that have very different activations, they have very thick white matter between them, then they're gonna start to coordinate, right? So the activation will flow in that direction. And so the matrix A usually is drawn from diffusion MRI data. It's like a fixed thing that we don't change. It can vary over subjects, and we do like to collect that if we can, because people do have subtleties in the differences in their white matter structures, which could inform how their brain activation patterns like move through the state space. So I think that A is not learned. It's like measured from the data itself, from either the subject or from an external set of subjects that we average and use as a placeholder for the structural connectome. But the linear versus nonlinear, it's like linear is simplistic, right? I know, and it does okay, but there's also like nonlinear approaches, but then the dimensionality of the model goes up, and then you're probably overfitting, and it's just like a huge, yeah. So there's a lot of active work in this area, actually. Yeah.
So you're saying that you can use the psych results to be able to treat patients that have, like, for example, that had a stroke. And one of the things you mentioned is that if they spend more time in this particular area where, for example, with the dominant hand, then they recover better. How exactly would one spend more time in a particular
Yeah, that's a good
state?
question. I love this question. So I thought about this. So there's something called transcranial magnetic stimulation. And depending on the frequency of the stimulation delivered, you can get hypo or hypoactivation of the region that's targeting. So right now, TMS is actually used in clinical trials for treatment of stroke. So people actually use now this TMS coil that activates neurons, so you can actually stimulate them and cause them to activate. So my idea was that if you could then use the TMS perhaps on someone who has a dominant hand CST and needs to spend more time in that FPN state, you could potentially stimulate the FPN area to try and upregulate that. There's also things like TDCS, which is direct current stimulation, which is like two big electrodes that kind of stimulate the entire brain, but you can put them in specific spots to get a specific pattern of activation. So you can there's ways that you can do non-invasive stimulation to guide the activation patterns.
Would these kind of studies be able to establish causality, or are they more associations?
Okay, yeah, that's a good question. So causality in fMRI, in brain activation studies, the best thing that I could say for that is either non-invasive stimulation. So you're actually injecting signal into the system and you look at what happens as a result. So that's causality. Or other types of non-invasive stimulation like visual stimuli or auditory stimuli or some external stimuli that actually would cause the brain to activate in those instances.
I mean, because this is kind of used for treatment strategies, and I'm just wondering what level of evidence would be required to make that.
Yeah, so for example, for the one I talked about, the TMS trial for stroke interventions, they have a sham stimulation where they put the coil up next to the person's head, and they do a subliminal threshold stimulation. So it's low enough so that the person feels like there's something happening, but it's not high enough to actually stimulate the neurons to get the effect that you think is coming from TMS. So there's ways that you can construct clinical trials that will allow you to get placebo effect versus actual effect of these strategies. It's a bigger problem with psychedelics. So that's a big thing right now. Everybody's talking about how do we do a placebo for a psychedelic. So the thing they've been doing mostly, the most recent ones I've seen, are low dose of psilocybin and then a high dose of psilocybin. So I guess the low dose is enough that they sort of feel something weird, but it's not enough that they have a perception hallucinations and things like that. but it's a big problem, yeah.
We're a couple of minutes past the nominal end time, and my kid has piano lessons, so. So.
If you still had a question, you can talk to me after.
Yeah, I suggest we take further questions with food and thank the speaker again.